Really broadly, we're going to be looking at the connection between mining and metabolism. So the work that we do in my lab is a combination of things that we call functional genomics. Genomics is studying all of the genes in an organism, as opposed to genetics would be studying an individual gene. And we combine that genomics with metabolomics, and so we're looking at lots of metabolites, hundreds, thousands of metabolites at a time, not individual metabolites. So we're taking these very broad system level approach to look at the connection between genetics, metabolism, genomics, metabolomics. We're going to pause for technical difficulties.
I think I might have stopped the interrupted the sharing of the screen. Just as you changed the slide, it did not change online.
Okay, we're back online.
We're good now.
All right, so what we do in my lab is combine these system level approaches of genetics, genomics, metabolomics. And what I want to do today is apply that to a mining environment. So by definition, any workplace has its associated stressors. And mining, by its very nature, has a physiological stress that's associated with mining. Part of that is simply the environment that miners are working in. And one of the interesting aspects of that is as you go deeper, the pressure in the mine gets higher and higher. And we have reason to believe that that pressure itself causes a physiological response. And what I'm doing in my lab with my research is trying to understand that physiological response to the change in environment so that we can make suggestions to minimize the negative impact of that change in environment. So how do we better enable miners to work in that high pressure, deep underground environment? The first part of that project is understanding how does metabolism change in response to simply that change in environment, or that simple change in environment. Really broadly in my lab, we look at the connection between genomics, or between genetics and biology. How do you go from gene sequences, how do you go from DNA sequences to interesting aspects of biology? And so even if you look at the simple selfie from the lab that I have here, there's a fair amount of diversity within that group. We're really good at geneticists, at sequencing DNA. We're really good at biologists, but also simply as human beings, at understanding or at least describing biology. Most of genetics and genomics at the moment is trying to connect between those. How do we go from DNA sequence to even the simplest changes in biology that we observe? Right? And so those are the kind of questions that we ask in my lab. We can apply that by saying, how does the miner respond to the environment in interesting and complex ways, in the same way that we talk about biology as being this sort of combination of complex phenotypes. Okay, so we're going to take this basic research and try to apply it into a mining environment. In my lab, we use the fruit fly as a model species. I have brought a fly, or brought a vial of flies. Let me see. So this is a vial of flies, and the reason I wanted to show this to you is this vial, if I take a step back, is about the size of maybe a big pack of gum, and that vial of flies will comfortably hold about 200 individuals. We usually put about 50 to 100 individuals in there, but we can put as many as 200 flies in that. All right, and we'll come back to why that's important. But so the fruit fly is a model system. A lot of the questions that we ask in my lab involve genetic diversity, so how similar or different are individuals? One of the reasons we work on flies is they're super easy to maintain in the lab, which is what we've done here. They're actually relatively easy to genetically manipulate. And so for the last 100 years, we've been engineering flies in one way or another, and we can use that experimental approach to understand basic biology. Other reasons that we work on flies are simply things like they're easy to grow, they're inexpensive to grow, and they're really tiny. A lot of biology is interesting in how individuals differ. If you put this in a mining context, if we want to understand the physiology of working deep underground, one of the really important things is understanding how do different people respond differently to that same environment. When we work with people, when we work with larger study animals, you're limited in the number of individuals you can work with. When we work in the fly system, an average experiment in my lab will have between 1,000 and 10,000 individuals in that experiment. And that gives us a huge amount of power to resolve really small differences. And it ends up a lot of biology is interesting in these subtle differences that are there. And having things like a large sample size, because you can literally take 1,000 flies underground, allows us to ask and answer some really interesting questions. Okay, so all of that depends on being able to connect flies and humans. And so fundamentally, when we work with any model organism, what we're doing is we're asking a question in one organism that we're really interested in answering in another organism. So if I want to understand minor physiology, why am I studying flies? One of the reasons is because we're all evolutionarily connected, there are enormous amount of similarities between flies and humans. And strikingly, the similarities are, the other way to put that, it is striking how similar we are as opposed to how distinct. So up to 80% of the genes that we study in humans are found in flies. But we can go in and experimentally manipulate flies in a way that we would never do in a human system. The genetics is up to 80% identical. In some cases, it's higher than that. The metabolism, the biochemistry, is even more similar than that. So when we understand the biochemistry, understand the metabolism in a fly, we can then associate that back to humans. It is not a perfect model. There are obviously differences between humans and flies, but it's striking the amount of similarity that really is there and how much we can tell about human metabolism, human physiology by studying that fly model. Okay, let me give you two quick examples of how this has been used in other systems as sort of a proof of principle as to how we're going to use this in a mining example. So obesity is a global problem in humans. Something like in North America, over 20%, and I think in the US, it's close to 30% of the population is clinically obese. Sudbury, from our local perspective, has the distinction of being the fattest municipality in Canada. We're also like the second happiest, so there might be a correlation between this. But obesity is a global problem, and it's actually sort of like mining. There are issues in mining that are global issues that have a local focus here in Sudbury. Obesity is a global issue that has a local focus here in Sudbury. We have actually done a lot of work in the fly system to understand obesity, and one of the biggest challenges from a health perspective with obesity is this strong correlation between obesity and heart disease. As our population ages and as this obesity epidemic continues, heart disease is going to become a bigger and bigger problem, a bigger and bigger load on our health system. In the fly system, we've actually been able to dissect out this connection between obesity and heart disease, and it's not as straightforward as you might think. You can take flies and feed them on a high-fat, high-sugar diet, like a sour cream glazed donut, one of the high points of Tim Hortons, and you can create obese flies. And those obese flies actually get heart disease. And we can follow that heart disease. Because these are flies, we've been able to experimentally manipulate the system and go in and explore that. And we found one of the genes that actually connects heart disease and obesity. And genetically manipulating that, we can create flies that become obese but miss that heart disease connection. And that gives us an idea of where we can look in the human system to try to treat that connection between obesity and heart disease. In a similar example, also using flies, we've looked at, and when I say we, I mean broadly in this sort of fly genetics world, we've looked at the connection between diet and lifespan. And so one of the things that was observed over 2 decades ago is if you restrict diet, you can actually extend the lifespan of many organisms, likely also humans, but certainly in model organisms. One of the interesting complications, though, in this diet restriction is that the kind of diet matters. And so when you take flies and you restrict the number of calories that they have access to in a reasonable point, you can actually extend the life of the fly. So you don't starve them until they die. You starve them until they're essentially thin and they live longer. They're not as happy, but they're longer lived. Interestingly, you can switch that around by changing the diet. And if when you restrict those calories, you also push the amount of protein that's in the diet, you reverse that trend. And you actually get shorter lived flies simply because of the ratio of protein to calories in the diet. And the reason that I bring this up is we've been able to show this really clearly in the fly system, and that this has implications for human health. This is exactly what I'm proposing that we do with the kind of research that we're doing in the mine environment. Once we understand how physiology, how metabolism changes in the mine, we can make simple dietary suggestions to limit the negative impact of working deep underground. Here's a deep underground environment. Here's the physiological response to that. These are the good aspects of that we want to promote. These are the negative aspects of that we want to downregulate. And here's how we can do that in a simple diet-based manner. Okay, when we look at metabolism, almost by definition, we're looking at a complex network. So I put this up here as just an example of just how complicated metabolism is. And this is obviously a simplification of what metabolism actually is in an organism. But it's all based on these interconnected webs of metabolites. It's not individual pathways, it's a web. And that's how it works. That's the biology of the system. The question then is, how do we study it? One of the ways that we do it in my lab is we break a piece of that out. And so rather than trying to study that entire page, we take a section of that and we work on individual pieces. And so in this case, we're studying a number of enzymes that have a shared function in what we call a metabolic cofactor. And so it's a piece of energy currency that's shared within the cell. And these four enzymes that I've got diagrammed here in red share that energy currency across them. And so what we've been doing in the lab is looking at the fundamental biology or metabolism that's driven by those connections. And we do that with a whole fly. And one of the somewhat surprising things is that this actually works. So what we're interested in is how metabolism changes in response to something, whether it's when we change the environment or whether we go in and genetically engineer the fly to change access of one enzyme or another enzyme. And we can actually do this really well using an entire fly. We take the fly, we put it in a tube, and we grind it up with a Dremel tool. We get a fly homogenate, a liquid fly. We spin the solids out of that homogenate, and that little liquid fly is then a really good biochemical model for the metabolism in that fly. This is important because one of the drawbacks to flies is they're really small. And if you had to do things on a tissue by tissue basis, the dissection of the digestive tract out of the fly is a challenge. It absolutely can be done, and there's some really interesting work that is done on a tissue by tissue basis. But the fact that we can answer some really interesting broad questions using a whole fly homogenate is really striking. And so we can take thousands of flies and really quickly process them through. We put them in these microplates that have got 96 wells, and suddenly you have almost 100 samples that you're working with, and that gives you a lot of investigative power to explore these different systems. And then we run those through with various assays. We look at enzyme kinetics. We look at the different amounts of metabolites. What we have been working on for the last 5 years is this metabolomics approach. And the idea behind metabolomics is rather than looking at 5, 10, 15 different metabolites, we can look at 2,000 metabolites at a given time. And the technique that we're using for this is called liquid chromatography mass spec. So we take our liquid fly homogenate, we run it through a liquid chromatography front end that separates that complex solution somewhat into its individual components based on retention on a chromatography column. And basically what it allows us to do is take this really complicated system solution and look at pieces of it through time. We run that liquid out of the chromatography through a mass spec. The mass spec ionizes the liquid, bounces those ions through a vacuum chamber, and gives us an accurate mass. The punchline is you start off with a liquid fly, and then you end up with a data set of 2,000 to 3,000 individual metabolites that we have a concentration for. And so what we're looking at is to say, in this fly, say that's been in a deep underground environment, compared to this fly that's on the surface, what proportion of those 2,000 metabolites that we can identify have changed in response to that environment. And we do this when we look at oxidative stress, when we look at differences in genetics. We can look at it simply as the fly ages through time. What this allows us to do is to start to go back and put together these complicated networks. And so now for each of these points, up to 2,000 points in a graph, we can say this is how these metabolites change in response to whatever the response. In this case, deep underground mining. That is the theory. The reality is at the moment we can identify about 100 metabolites. We can resolve between 2,000 and 3,000 putative metabolites, and a lot of the work that we're doing now, and a lot of the work that we propose to be doing over the next 5 years, is putting names on those metabolites. So metabolite X changes in response to deep underground mining. What is metabolite X? And then how do we integrate that into this idea of a metabolic network to understand how metabolism, how physiology is changing? So why flies? This entire project, this use of flies in the SNOLAB environment to study pressure, came out of an interesting observation by one of my colleagues. So Amanda Merritt is professor at Western University. And she was doing a series of really elegant experiments looking at mating preference in flies. And it's a really good system for looking at the genetics of distinguishing individual species. So how does one species separate from another species? Part of these experiments are how frequently flies mate. And to put that differently, the experiments require the flies to mate. Amanda and one of her undergrads made an interesting observation that at some times the matings did not work. And so they would put male and female flies together that they expected to produce offspring 100% of the time, and they wouldn't get offspring. And it was a really sort of striking observation. And because they're really good observational biologists, they took a step back and they started looking to see if there was a pattern. And what they found was that it appeared that when the weather was changing, the flies were less likely to mate. And so they took a step back and said, okay, if that's actually true, what could be driving this change in behavior? And they thought it might be atmospheric pressure. And so they put the flies in wind tunnels, a wind tunnel, and changed the atmospheric pressure in a very predictable way and showed that you could increase or decrease the mating frequency of these flies, and wrote a really elegant paper around that. And I read this paper at about the same time that I started getting tours of SNOLAB. And the punchline of this is if the flies are able to change their behavior in response to pressure, there has to be some sort of metabolic signal between pressure and behavior. And so about this time, I started getting tours of SNOLAB. And so SNOLAB is a clean lab facility located 2 kilometers underground in the Crichton mine. And so you take the cage down 2 kilometers, you walk a kilometer through the drift, and then you leave the mine environment, and you come into the environment that I've shown here, the absolute clean lab, complete dust-free environment. And there's amazing research going on in SNOLAB. They won the Nobel Prize in 2015 for work on particle physics that was done in SNOLAB. So I was getting tours of SNOLAB simply because it's an amazing scientific facility, but when you get these tours, you're told to be cautious when you go back up to the surface because you're going to be more tired having spent the day in this deep underground environment than if you'd been on the surface. And I put together this with Amanda's observation of this change in behavior. If you're actually more tired from being underground, how is your physiology responding to that? And since we know from Amanda's work that the flies will actually respond to pressure, can we use the flies to understand how metabolism changes with that shift underground? And so we ran a pilot experiment a couple of years ago where we took a set of flies and essentially put the flies through a single workday in the mine. They went down in the 6 a.m. cage, they came up in the 4 p.m. cage, we flash froze them on liquid nitrogen, and then ran them through that liquid chromatography mass spec that I showed you in the previous slide. And what we found was that a single 10-hour shift 2 kilometers underground changes about 15% of the metabolites in the fly. Right? So the metabolism is changing. For context, a sublethal dose of insecticide changes about 30% of the metabolism, 20 to 30% of the metabolism. So what we can define as a really harsh environmental change, insecticide exposure to or an insect exposure to insecticide, gives you about twice the response that you get from a single shift underground. And that's suggesting that the fly physiology is changing profoundly in response to that movement deep underground. And that suggests that human physiology is changing as well. There are a lot of aspects of this. So one of the things is this is a single exposure, and it's very likely that through time, that response is going to change. And so what I'll show you is the ways that we're trying to look at that, where we look at single exposures, we look at multiple exposures, and we look at differences in exposure through time. But the punchline from this piece is that the fly absolutely responds to being deep underground, and suggesting that miners also respond to being deep underground, that we have an opportunity with the fly model to piece together exactly how metabolism is changing, and then use that understanding of metabolism to make suggestions to say these are ways that we can make the mining environment healthier by promoting the good responses to being under high pressure and down-regulating or decreasing the bad responses. Okay, I've said pressure. We don't know that it's pressure. We know that when flies are underground in SNOLAB, they respond in a metabolic fashion. So their metabolism changes to that exposure. The most likely environmental feature is pressure because SNOLAB is temperature controlled. And flies, one of the ways that flies are not a great model for humans is flies are cold-blooded. They're endotherms, ectotherms. So they don't thermoregulate. Right? So one of the things that we cannot study in the fly model is thermoregulation. So for that reason, SNOLAB is a great environment for us because it is actually thermoregulated. So in a case where we have not changed the temperature, but we have changed the pressure, we're getting this physiological response. If this is in fact pressure, we should be able to simply change the pressure and mimic that. And so we have a companion study where we're using a pressure chamber in a lab here in Sudbury to see if we can duplicate the response that we see in SNOLAB. One of the interesting frustrations or challenges is that it is really hard to get a pressure chamber that's as consistent as SNOLAB. So 2 kilometers underground, the atmospheric pressure is between 22-ish% higher than it is on the surface, and it is dead solid at 22% higher than the surface. When we've tried to mimic that with the pressure chambers, we don't get that same consistency, and we get it for a much shorter period of time. So we're still trying to see if we can replicate these studies in a more controlled system. So we've been looking at the metabolism. The differences that we see in the metabolome should be a response to changes in protein activity, changes in gene activity. And so one of the other projects that we've been working on for the last just the last couple of months is to also incorporate in gene expression. So in humans, we've got about 22,000 genes. In flies, they've got about 15,000 genes. In any given cell, in any given organism, only about 10%, somewhere between 5 and 10% of those genes are actually turned on at a given time. The way your body responds to anything in the environment is not a matter of the presence or absence of genes. It's a matter of genes being turned on or off. And so one of the ways that we can look to see how organisms respond to the environment is to look at gene expression. And so what we should be able to do with these studies is now say, here's how metabolism changes. Here are the changes in genes that are driving that, and how do we connect those two together? And that'll give us a better idea of how the miner is responding to being deep underground. And so we've started doing these gene expression studies with Heath McMillan, who's a new researcher at Carleton University, and we think they're going to give us a really good insight into the biology of being deep underground, not just the metabolism of deep underground, but a more complete biological picture. All right, I mentioned at the beginning that one of the things that I'm looking for is feedback about the kind of questions that we should be asking. So when I first started presenting this data to SNOLAB, to the researchers who were working in SNOLAB, and explaining to them we were studying metabolism and this metabolic impact of being deep underground, a number of the researchers came up and said, one of the things that we've noticed working in SNOLAB is that we have difficulty, we think, making decisions deep underground. And that if you're working on an experiment, you've got something going on, you're trying to decide whether you're going to do this or do this, making that decision seems to be harder to do deep underground than it is in the surface lab. And so we started thinking about that, and there are a number of reasons why it could be harder to make a decision underground. The researchers are well aware that they're in a $500 million facility. Any given experiment can be $30 to $50 million. If you flick the wrong switch, you could in fact compromise a $30 million experiment. So maybe the difficulty in cognition is simply that sort of intimate exposure or knowledge of the fact that if this is wrong, you could be compromising 10 years of response. Or it could simply be that you're deep underground, and that physiological response to being deep underground makes it harder to make a decision in the same way that being really tired makes it hard to make a decision or be under some sort of physical stress. So we can actually test that in flies. So we can do experiments with the flies where we task the fly with a particular decision or a particular learning trial and look to see how the flies respond to that learning trial on the surface versus underground. The flies don't know that they're in a $500 million facility, and so they're not going to be impacted by this, the psychology of this. They're going to be impacted by the physiology. The challenge that we have with these learning trials in the fly, or memory trial in the fly, is that most of them are actually based on mating behavior. And we know from Amanda Marion's work that has started this whole project that mating behavior absolutely responds to pressure. And so that's not going to give us the answer to the question of whether this pressure is changing decision ability, and that the flies seem to be modifying that decision based simply on the pressure. And so we're working now with some other trials where you can actually teach flies to eat particular foods and avoid particular foods. And so one of the ways that we're looking at doing this is, can we actually change the ability of the fly to remember what the good food is in the mining environment. So the idea here is we have an observation from the end user saying, when we're underground, this is how we think we respond. Is there any biological reason behind that? And so we're looking to see whether cognition is impacted simply by the high pressure environment of being underground. And if there is, then we can make recommendations to the SNOLAB users. Don't make high pressure decisions while you're underground. Okay, so one last study. So one of the things that's come out of this, especially with the changes in metabolism, because the underground environment seems to be having a profound effect on the metabolism of the organism, a really reasonable question is it actually shortening the lifespan of the organism? And one of the advantages of working with flies is the lifespan of the fly is relatively short. And so we've just wrapped up an experiment where we actually looked at this. We took a set of flies, and I've forgotten exactly how many flies, but we're probably somewhere between 400 and 800 flies. And we looked at the lifespan of these flies in the surface lab versus flies that went in and out of SNOLAB on a daily basis. And one of the things that I haven't mentioned, but that our impression of these changes in metabolism is that the flies are responding to the change, not the absolute pressure. And so it's the fact that you're going from one pressure to a higher pressure, back up to the ambient pressure and back down. It's that moving up and down of pressure that's actually leading to this physiological response. So we didn't just leave the flies in SNOLAB. We brought the flies up and down every day, and they worked on a Monday to Friday work schedule. And you can see in this slide the results. Since this is the only, these are the results that we have. Let's see, does my cursor show up? No. Okay, so if you look at the data on, I don't know if it's left or right, but if you look at the data curve, one's a picture of a mine, so that's not the data. The other is a curve, that's the data. And what we're looking at is two sets of lines, and one is slightly to the left and the right. Through time is along the x-axis, and the percentage of flies that are still alive is on the y-axis. And so if you start on the left, 100% of the flies are alive. And if you move to the right, all the flies are dead. And what we're following in that curve is the die-off of flies through time. And the reason that there's sort of two sets of curves here are there males and females. Right? And so we took both males and females underground and we observed those. One of the punchlines is that if you look at the far end of the graph, all of the flies die at about the same time. So the ultimate lifespan or longevity of the flies is very similar. And so one of the reassuring punchlines is, although mining seems to be changed, or exposure to the deep underground environment is changing the physiology of the fly, it's not shortening the lifespan of the fly. And so we're not immediately saying that mining is an occupation that could be shortening your lifespan. What we're saying is mining is an occupation that seems to be changing your physiology. Can we limit those changes? Right. One of the reassuring pieces of this data set is this separation of the two sets of lines. And so there's a set of lines to the left and a set of lines to the right. Those lines represent surface and underground, and the two separations are male and female. And what we see very strikingly is this group on the left that are the males, and the males die off much faster than the females. And this is reassuring because every study of longevity in flies shows that males live a shorter period of time than the female. So we're able to resolve or replicate this known phenomenon. Males are shorter lived than females, and at the same time don't see a separation based on surface versus underground. Now, interestingly, just at the inflection point where the flies start to die, there is a suggestion that the flies that are going underground start to die a little bit faster. And that would be consistent with this change in metabolism that we're seeing, that there really is a physiological response, but that overall, when you look across all 800 flies in the study, you don't see a large reduction in longevity. So is there a change in metabolism? Everything suggests there is. Does that result in a reduced lifespan? Overall, it doesn't seem to. Okay, the last piece of the study is that generally when we work on flies, we literally look at flies in vials like this. And the vials are sitting on the desktop, and the flies are relatively inactive. They sort of walk around. Mining and many occupations are inherently active. And so one of the things that we wanted to incorporate into the system was activity. And so to do that, we put the flies on a treadmill. And so this is an image of a fly treadmill. The fly vials go on the treadmill. And I don't think I can turn the sound off. I'm hoping that you all get stuck with that. Oh. Okay, so yeah, so it's actually, so the question from the audience is, are they flying? Only if you throw them in the air. So they don't actually, but a lot of times flies actually walk. And so I know they're called flies, but it's because walk doesn't work as a species name. No? Nobody? Okay. So they're not flying, they're walking, but in a pilot experiment. So then the question is, okay, they're not flying, they're walking. Is this actually is this legitimately exercise? Are we changing the way that the flies can we change the metabolism of flies simply through exercise? So the bottom figure on this is actually data from an undergraduate thesis from last year. And what this student could show was if we look at the metabolome, so we look at these 2,000 metabolites for these flies, and we can graph it across principal components, which is just a way to separate out the data, you see a very clear separation of flies that are inactive versus flies that are active. And so in as little as 15 minutes of essentially gentle walking on the treadmill, we can change the physiology of the fly. And so the set of experiments we're running in SNOLAB at the moment are to look at flies that are on the surface, flies that are underground, and in both of those locations, flies that are either relatively inactive or are gently active. And the idea with this treadmill assay is to get the flies, to promote the flies to walk sort of gently. And what we do is we have a 2-hour exercise period, and they exercise for 15 minutes. They get 5 minutes off, 15 minutes on, 5 minutes off for that entire 2 hours. And we do that once a day. And we've actually developed the treadmill assay because of the SNOLAB project and looking at the effects of active physiology underground. But I have a new student in the lab who's doing her 4th year thesis now and is coming back to do her master's, who's actually looking at using this same system, but to look at insulin resistance. And so flies are actually a good model for human diabetes. And she's looking to see if we can limit the impact of poor diet, high sucrose diet, by simply increasing the exercise of the fly. She's just started that project now. But we've got really interesting indications that a limited amount of exercise significantly changes the metabolism of the fly. Okay, and so we are incorporating in with this treadmill the idea of active exercise of the flies. And you can actually see the difference in fly activity. So the flies that are on the desktop are much less active than the flies that are in the treadmill. Like any of us on a treadmill, they get really tired of the treadmill and they stop walking and they just sit there and spiral around the sides. The video that I used to have was our first treadmill and it went too fast. And in whitewater kayaking, there's a thing called getting maytagged, where you get stuck in a vortex and you just get spun around, right? And so these flies couldn't actually keep up with the pace of the treadmill. And so you turn it on and they would spin around and the flies would just bounce off the walls. They were super active, but not in a healthy way, right? And so what we had to do with the treadmill was scale the pace of that treadmill to a way to keep the flies active. To keep them engaged, we reverse the direction of the treadmill. And so they go 30 seconds in one direction, and then they go 30 seconds in the other direction. So in your own exercise, what you may find is if the treadmill is just not doing it for you, after 30 seconds, just turn around and try to walk backwards and see if this doesn't engage you in your treadmill. It works, and the argument, I guess, is it works for flies. Maybe it'll work for humans. So long term, what we're trying to do is take this fly system for the reasons that I told you at the beginning of the talk and use it to understand the metabolism and physiology of working deep underground. Because it's the fly system, we can do things in a relatively short period of time. That 1 week exposure underground is similar to an entire lifetime of mining exposure for a human. Right? And so we are able to do a study that would take decades in the space of literally 5 days. We can do it with thousands of individuals. Trying to collect data from 1,000 humans, 1,000 miners, is an enormous task. It's a very simple task to do that with 1,000 flies. And then in addition to that, we can go in and use the powers of flies and model species to do things like genetically engineer, set up specific crosses, change the diet. And so we've got this combination of a really great field site in SNOLAB with diet studies, with genetic studies, all of which will give us a better idea of what's happening with the metabolism and the physiology of the system. And then the ultimate goal is to then go back and make recommendations and say, for example, if we find that protein metabolism is downregulated, then a high protein diet isn't consistent with working deep underground. Or if fat metabolism is upregulated, then a different balance between fat and protein in diet would make sense to make mining a healthier workplace. All right, let me finish with a couple of quick acknowledgments. The people that actually did this work, the exercise and diabetes work is being done by Michelle Ng at the moment. John Hick in the center is a graduate student who's doing the SNOLAB work. All of the metabolism work is a joint effort across the lab, and all the graduate students that are shown here have been a part of that. My daughter helps me collect flies, and she's really cute. So I put a picture of her up as well. And then funding agencies, the Natural Sciences Engineering Research Council funds a lot of this research. Most of my funding comes from the Canada Research Chair, which gives me the flexibility to ask really interesting questions, such as, can I study the metabolism of working deep underground using the fly system? All right, with that, let me see if there are any questions.
It's really interesting. You answered one of mine. I was going to ask, what is a 10-hour shift equivalent in terms of the lifespan of a fly? And so you answered that, and then it made me think, is there opportunity in the future to kind of replicate what you were talking about, so taking the flies up, and then down and up and up and down, so that it's more like, you know, they've done their shift work where they're coming and going, because as you said, it's the pressure change.
Yeah, okay, so there are two parts to your question. One is, do we have an idea for what the biological equivalent of the 10-hour shift that they're on is? And then two, are there ways that we could try to make the exposure that we do have more consistent with what a miner might experience? It's really hard to say. SNOLAB is an incredible space to work with. Vale's contribution in kind to SNOLAB is astronomical. The amount of resources that go into maintaining that. The limitation is we work within the shift schedules that are there, so we work on that shift schedule. I think once we get a better idea of where we see changes, and one of the experiments that we need to do is leave the flies down long term, and do we see a different response than flies that are going up and down? We sort of have a hierarchy of the kind of questions that we're going to ask. Once we've done that experiment to compare multiple day exposures to a full day, multiple down ups with a week of down, then we can make arguments and say, all right, well, how often can we bring flies up and down, and can we get a response? I mean, even working within the shift schedules that we have now, if we had flies that were going up and down as often as possible, that might give us a better idea of how that change actually impacts the fly. It's a really great question. In any of these things, trying to extrapolate from a short-lived organism to a long-lived organism is going to be a guessing game. And we basically just have to defend the guesses that we make. Yeah.
Opposite string.
Elephants.
Are you familiar with my research on space?
Yeah.
And are they doing any research there that's similar to yours, but just at the opposite extreme?
Yeah, so the question is, am I familiar with the work that's being done on the International Field Station with flies? I know that they have flies going up to the International Field Station. I don't remember what the question is that they're asking. Yeah.
Changes in physiology as well, and those going to be the opposite of what you find down.
Yeah, so the question is whether they're opposite because you're going to the space station. I don't know what the pressure is at the space station. I think it's actually, I mean, I know that it's regulated. One of the questions that I've had from the mining community is that mines are very much going deeper down and into higher and higher pressures, but they're also going higher up. And so there are a lot of mines that are now starting at high altitude and then moving down into the earth from there. And we very much could ask similar questions using those high altitude locations. I tried really briefly to pitch to one of the airlines that they should let me put my flies on one of their planes and have it go up and down. In that case, we know that it is the opposite effect.